In the laboratory, we work with some proteins which are known as protein kinases. These proteins has as a task the modification of all the proteins in the cells. And this is very important because then the cells know how to respond to things that happen outside the cells or things that happen to cells that are neighbor to them. The proteins kinases that we are working with are named DIRCs, and they are a family and they are very similar. But one of the members of the family is very important because when something is going wrong with this protein, disease happens in humans. So we want to understand how this protein works in normal conditions, and then we think that this would help us to understand what happens when the protein is not working normal in those cells that are under disease conditions.
With our work, we discover a novel function of this enzyme, DYRK1A, within the nuclear compartment. So within the nucleus, the genes are expressed through the action of a huge macromolecular complex, which is called RNA polymerase II holoenzyme. The activity of this RNA polymerase II has to be regulated for the cells to have a proper function and proper life. So what we discover is that DYRK1A, our kinase, is able to go to the DNA in which the RNA polymerase II is present, and it activates this enzyme by the modification of this enzyme through the phosphorylations, as Susanna said, but adding phosphate groups on the RNA polymerase II, and these modifications renders this enzyme active and able to transcribe several target genes.
For the group, this publication that Chiara has explained has been very important because for many years we've been working with this protein kinase, DYRK1A, on activities that are related with the cytoplasm of the cells. And this is the first indication of the protein kinase having activities in the nucleus. This is very important, not only for us, because it has opened a new avenues of research for the group, but also for the scientific community, because now think in other ways of connecting the activity of the kinase with disease in humans.